we're trying to copy DNA, and this is kind of shows you everything that happens. If you can understand this, you pretty much understand the process. But I'm going to show it to you with the magnets here, and pay good attention because I'm going to make you do it with these magnets. So the first thing that happens is there's a couple of enzymes that unzip the DNA. One of them is called DNAligase. It goes down and opens it up. And the other is called topoisomerase. There it is, topoisomerase. And it goes in front of the helicase and prevents supercoiling. Supercoiling sounds like a cool term, doesn't it?
Yeah.
I want to show it to you, how supercoiling works. If I take a couple of strings, Dylan, are you with me? If I take a couple of strings and kind of wind them around one another, imagine that's DNA. And if I'm going to take my mask off for this. Because I don't have 3 arms. If I try to pull this apart, watch what happens. That's supercoiling. See how tight it is there? It'll eventually bend. Look at what it does. Bends. See that? That's supercoiling. You can't have that happen. You can't just separate DNA without running into supercoiling issues. So, the topoisomerase goes in front, and it'll actually, to prevent this supercoiling at the end, it'll actually snip the DNA and reattach it and relieve the stress that builds up. Isn't that a cool enzyme? By the way, these enzymes that I draw, they're always way bigger than my magnets. This DNA helicase is a big thing like this. And the topoisomerase is a big thing. It kind of wraps around the whole DNA and manipulates it. But if my magnets were that big, they wouldn't fit in my little folders. Okay. Oh, put the mask back on. Let's get a shield. Y'all want to get a shield?
Yes.
Huh? You want to get a shield?
Why not?
Shield us. Shield. No way a virus could get by this. It does help them. I'm not an antivirus, fake virus guy. All right. The DNA the DNA helicase unzips the DNA. The topoisomerase prevents supercoiling. And then, I messed up yesterday. I forgot about DNA primase. There's an enzyme that comes in first called DNA primase. And it makes what's called a primer. An RNA primer. And what it does, it takes RNA nucleotides. Now remember, RNA nucleotides have U instead of T. Y'all remember that? DNA nucleotides are ATGC. ATGC. RNA nucleotides are AUGC. AUGC. So, they're a little bit different. They also have ribose instead of deoxyribose. And you have to add a primer to the DNA before you can copy it. You have to have an RNA primer. So, the DNA primase takes RNA nucleotides and it adds them to the DNA chain. I'm just going to add a couple here. I don't want to make my primers too big. But in actuality, the primer might be, I don't know, like 20 nucleotides long.
Wait, this is after it says helicase goes through it, right?
The helicase has unzipped it already, so it's unzipped. Now the DNA primase puts little RNA primers on, and it'll also do it on the other side. But on this side, it has to go from bottom up, because new nucleotides can only be put on in the 5' to 3' direction. So, remember the phosphate is at the 5' end? Y'all remember that? So, notice we go here, the nucleotides are going from 5' to 3' in that direction. Here, the new nucleotides are also going from 5' to 3' in that direction. New nucleotides can only go on in the 5' to 3' direction. Important point. It makes it so this side can be copied continuously going down, but this side has to be copied in fragments. They're called Okazaki fragments. You remember that from regular bio? We make a fragment going up, we unzip it some more with DNA helicase, and we make another fragment going up. We unzip it some more with DNA helicase, and we make another fragment going up. That's on this side. But on this side, you're unzipping and just continually adding new nucleotides behind it. This side is continuously copied. You don't need the fragments. We call this side the leading strand. And we call this side the lagging strand. Why is it called the lagging strand? It lags a little behind. It takes a little longer to make the fragments. Because you have to make a bunch of fragments and then bond them together with special enzymes that I'm going to show you in a second. Are we okay right now?
So what are the directions of the previous nucleotides? What's the leading strand direction? I thought the leading strand was 3 to 5.
Well, if you look at the leading strand itself, it's got the 3' here and the 5' down here.
Okay. What about the lagging strand?
But the new nucleotides are always put on in the 5' to 3'. Right?
What's the lagging strand?
So the lagging strand, this end is 5' and this end is 3'. But the new nucleotides are always put on in the 5' to 3', so they go backwards. Other questions?
Is it because of the phosphates that they go the 5 to 3' because they take the energy or something?
New nucleotides are always added by, here we go. Do you see that OH that's sticking off the 3'? That is where the new bond forms with the next nucleotide. So that OH is removed and an H from the other molecule to make H2O. Do y'all remember those types of bonds? It's a covalent bond, that's right. What just happened? This is crazy.
Oh, no. What is this?
It's a big X. So that OH is where we form the next bond, and it's at the 3' end. So that's why we have to go 5' to 3'. So here's the first nucleotide in place. The next nucleotide has to be, that OH has to be taken off the 3' end, and you can attach like that and make that bond there. Between the phosphate group here, the phosphate group will lose a hydrogen, and that bond will form between the phosphate and the next 3' carbon. You'll get that bond. I think it's called a phosphodiester bond.
Yeah.
Anyway, that's why we have to go in the 5' to 3' direction. And a little extra knowledge. Remember the terminator bases from Sanger sequencing?
Yeah.
The reason why they're terminator bases is they've just removed this OH. They've taken that off there. I don't know how they do it, but scientists figured out a way to remove that OH. And now you can't make that bond. That's why it's a terminator base. Yes?
I remember reading something in the chapter. It said like how they take energy from the 3 nucleotides or something. Like, because you have to have energy to add it, like the polymerase does.
ATP?
Yeah. Does it use ATP?
No,
I don't know. Yeah, you probably do have to have energy to attach that. It's probably ATP, I would imagine.
Yeah, it says energies obtained from the nucleotides that have 3 phosphates, similar to ATP.
Okay. But it's something else.
Yeah.
Interesting.
I don't know what it is.
Interesting. See, I learn new stuff all the time, even though I'm old. You can teach an old dog new tricks. This old dog just learned something. All right. Thank you, Josie, for teaching. All right. We've added the primers. Are y'all ready to keep going?
Super ready.
Okay, so we're done with this enzyme. Now we got another enzyme, DNA polymerase 3. Why is it not DNA polymerase 1 that does the first thing? I don't know. Anyway, DNA polymerase 3 will come in and will take DNA nucleotides and add them one by one to the primer. And so we just click, we're adding new nucleotides. If that's a G, what goes next to it? A C. Way to all say it out loud together.
C.
If that,
C.
ah,
Oh. Like my grandma.
if that's a C, what goes with it?
J.
G. If that's a G, what goes with it?
C.
C. And we just keep going down the line. You see, on the leading strand, as we're unzipping, we can just keep adding new nucleotides. I'll do a couple more. There's a C. We'll add a G. And we just continuously make this strand. Now, what happens on this side? Well, we're going to go up in the opposite direction. I'm running out of G's. Click. And we're just going to go up, and we're going to keep going up. And there's actually a whole bunch of these DNA polymerases doing this at once, and they do it very fast. They add up to 15 nucleotides per second, I think, is the pace here. So they're going much faster than me. And
I thought it was 1,000.
It says a replication moves at a rate of 1,000 per
1,000 per
second.
second? I think that's in prokaryotes. But yeah, it could go even faster in prokaryotes. I think it's slower in eukaryotes.
Makes sense.
But gosh, there you're teaching me more stuff. That's really fast, y'all. So, even at 1,000 nucleotides per second, the whole DNA strand, even in a bacterium, still takes 20 minutes to do, to copy. That's how long, that's how, even 1,000 per second, it takes 1,000 seconds to do 1,000,000, right? Big championship math stuff there. And how long is 1,000 seconds? How many minutes?
42.
Really?
Wait, give me a second.
Real?
Wait,
1,000 divided by 60.
I got you. That's not.
It's about 20 minutes, I think. 18 minutes.
It is 16.6666 seconds.
Okay, there you go. 17 minutes. So, that's to do 1,000,000. But then you can speed the process up if you put more enzymes in. Okay, so are y'all with me here? We're gonna unzip some more and finish this thing up. And like I said, on this side, you can just continuously go, just keep going. And this DNA polymerase 3 will just keep going. But on the other side, I'm running out of nucleotides.
What if that happens in real life?
Then you haven't, if it's your cells, you haven't eaten enough. Your cells will, you get all of your nucleotides from your food that you eat. When you eat food, you're eating cells from another organism, right? Even if you eat plants. You're eating cells from another organism that have DNA, and you digest the food and write down their DNA and you use their nucleotides to copy your DNA. So that's why you eat, to get enough of these. So down here, here's the point I wanted to make, is we have to put, DNA primase has to come in now and put another primer down here. Put another primer down here. And then DNA polymerase 3 comes in, and remember we got to go up in this direction, and copies the DNA again, going up, and we're going to make it, got just enough nucleotides. So now we've gotten two sides. Now what has to be done? We're not done. We haven't made two copies yet, have we?
Zip it.
Well, it's already been unzipped.
I meant. Yeah, I don't know what I meant.
What's wrong? Why isn't this an exact copy of this? Look at it.
It's got RNA.
It's got RNA in it. We got to remove the RNA. So there's an enzyme called exonuclease. Let me go back to the picture here. Oh, there we go. There's an enzyme called exonuclease. Oh, they don't show the exonuclease. Anyway, what it does, it cuts out the primers. Snip, remove. Comes over here, snip, remove, and down here, snip, remove. So the primers are gone. And then we have to have another enzyme called DNA polymerase 1. I guess it's the first one they discovered, is why they named it that. That comes in and puts and replaces the primers with actual DNA nucleotides. Click. Click. And we got to replace this primer down here. Replace and replace. We're still not done. After you replace a primer, you have to tie it in. You have to make the covalent bond on the side. You can't just put, if we just put a primer in, it won't form the covalent bonds by itself. If we just replace the primer, you won't form these two bonds that tie the primer in on either side. So what you have to do is there's a final enzyme called DNAligase that makes those covalent bonds form on the sides. Ties that in, and it basically ties this in. DNAligase is an enzyme that ties pieces of DNA together by making covalent bonds between the sugars and the phosphates on the side. Those are all the enzymes you have to know. There's a bunch of them. And look, we got two equal copies. That's what happens in S phase of the cell cycle. Remember in S phase, you learn you copy the DNA. That's how it happens.
Whoa!
Yay!
Yeah!
Do you want to watch video footage?
Sure.
There's some good video footage here. Good video footage.
But this just happened. So there's nothing making this happen. Just
a lot of things making this happen.
No, just come for your actions that just happen.
But they don't just happen.
You much do.
They don't.
Hey, don't argue about whether they
New molecules of DNA must preserve the fidelity of the base sequence. DNA replication begins when enzymes break the hydrogen bonding between bases and separate the strands.
Try to watch the video.
Different enzymes then bind at the replication fork and extend the strand separation, opening a region to serve as template for the new strands. Polymerase enzymes then construct strands by adding units called nucleotides, which contain sugar, phosphate, and base. Each new base matches the template according to the base pairing rules. In this way, two new molecules of double-stranded DNA are made. Each DNA molecule contains one new and one old strand.
That's satisfying.
Did you see the fragments on this side?
Are they lagging?
That's the lagging strand. This is the leading strand on the top, which just goes continuously.
What fills it in?
What fills it in?
Yeah, you see those gaps? in the-
Here?
Yeah.
This is the DNA polymerase enzyme. It's going to keep going until it reaches there.
Will it
This
break
one's going
off?
to keep going until it reaches there.
That's it.
They aren't showing the primers here. Primers have to be removed.
So the lagging strand can have a bunch of them?
Yes. The lagging strand will have many polymerases going.
Why isn't it just, why does it lag?
Why does it what?
Never
Why is there fragments on this side? Is that what you're asking?
Yeah.
Because it has to, the new nucleotides, it has evolved, so it can only be put on in the 5' to 3' direction. So this has to go, they have to go backwards. And if you're unzipping this way, there's no way this side can go that way. You see, it has to go that way. So if you're unzipping in that direction, you're going to have one side that has to go backwards. And so you unzip, and then you start copying. Then you unzip some more, and then you start copying, and you'll end up with fragments.
Nice.
Do you want to?
Nice.
Wait, I'm not done. I have another video.
What?
More videos for you.
MVF. This is a better one. This is the best one.
Nice. All right, yeah, but
I think it's English people.
DNA is a molecule made up of two strands, twisted around each other in a double helix
They make
shape.
the best videos.
Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNAligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA
replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Wasn't that good? That was fantastic. Best video I've ever seen on DNA replication.
Is that new?
It is somewhat new. It's been made in the last 4 or 5 years, I think. All right, why are you cleaning up? There's no cleaning up to do.
Yeah, you're going to do this part.
We still got more stuff.
There's something DNA replication to do.
Okay, there's a little bit of a problem. At the very end of the chromosomes are areas called telomeres. Have you ever heard of telomeres?
Oh, don't they like run out at a certain...
Yeah, telomeres are areas of the DNA that are just like repeated sequences that really have no meaning. I don't know, it might be 1,000 bases long and just AATT, AATT, AATT. We call those telomeres. Every time a replication is done, the enzyme that makes the primer attaches and then starts copying. And what ends up happening is the new copy doesn't have the very end of it on the new copy. And so every time you copy, your chromosomes actually get a little shorter. But they have this long nonsense sequence so that the enzyme that first starts the whole thing has somewhere to attach and then copy. But the new copy is just a little shorter. And now it has somewhere to attach and copy, and the new copy is a little shorter. And eventually it gets so short that it starts running into part of the chromosome that is necessary information on it. And so this is what happens as you age. Your telomeres get shorter. They can actually tell how old you are by looking at the chromosomes and seeing how long the telomeres are. And you can do this. You can send in your DNA to a company. They'll look at the DNA. They'll look how long the telomeres are, and they'll tell you basically how much time you got left. That interesting? So even though I'm 52, I don't know because I've never done it. I might have the telomeres of a 35-year-old.
Probably.
And so that would think that I would have a lot more time. I might send it back and they go, you have the telomeres of a 70-year-old, which would mean I have less time. It's not an exact science, but there is a relationship. They do kind of know that every division, your telomeres get shorter, and then the cells can't divide anymore. Yeah.
Does everyone's telomeres differ?
Yes. Your telomere length does differ genetically. So your family might have longer telomeres. My family might have shorter. Now, there are some cells, like cancer cells, and stem cells. Have you ever heard of stem cells?
Yes.
Yeah.
That have found a way around having the telomeres shorten every copy. Yeah.
What's like the science behind like telomeres relating to your life, your lifespan?
Well, if the chromosome gets shorter every time you copy it, eventually, like I said, there's nonsense information on the end. Eventually, you're going to hit some important information, and at that point, you're ruining the cell by copying it. So that can lead to problems, of course. The DNA, if you can't, you're missing pages out of your instruction book that make the cell. You're going to screw up your cells.
Are there like diseases where telomeres are like shorter?
There's cancer. Have you ever heard of cancer?
Yeah.
that is a disease where the cells have activated this enzyme called telomerase. And what telomerase does is it adds a little segment on the end, a little extra segment. Okay, here's the normal end of the chromosome. Here's the telomerase enzyme. And when the telomerase attaches to the end, it adds a little segment of DNA at the end. And then it can start the division. And then you don't run into this problem of shortening the telomeres with each division. It's a special enzyme. It's called telomerase. For some reason, it's active in cancer cells and keeps cancer cells multiplying over and over without dying. Have you ever heard of HeLa cells?
Yeah, I had to read a book about it.
Henrietta Lacks was a lady whose cells are still being used today for cancer research. There she is. And there's her cells. She had ovarian cancer. And they took a copy of her cells, and they're still multiplying today because they got this telomerase enzyme that adds every time. It doesn't shorten it every time. It adds a little segment before it does the copy. And for some reason, cancer cells just can keep surviving. They don't die out. And in your stem cells, have you ever heard of stem cell research? They have the telomerase enzyme activated. And so they stay young. They don't ever get old. And you can you have stem cells in your body that can replace dead cells, and they remain young. So if we can figure out, if you've ever heard of stem cell research, it's researching how these cells do things. If we can figure that out, that might be the key to keeping you young. Yes.
Don't we have a lot of stem cells when you're born, or is that
the first, when you're an embryo, all your cells are stem cells, pretty much. Even before the embryo stage, when you're just maybe 100 cells in size. They're all stem cells.
I saw something like your umbilical cord and blood has all this stem cells.
Yeah, right, yeah.
If you say that you can use it.
A stem cell is a cell that can become any cell. So what they do is when you're born, they take a lot of those stem cells, freeze them, and let's say one day you get paralyzed in a car wreck. Maybe they could take some of those frozen stem cells from when you were born and inject them where your spinal cord broke, and maybe they would grow into new spinal cord cells and cure the paralysis. This has actually been done. Stem cell research has cured paralysis in lab mice and rats and stuff, and they're even trying it on people, and it has had some success. So that might be the new thing, stem cell research. Okay, so the next part of the book, you see this person right here. He's got xeroderma pigmentosa. And these are all like badly damaged cells from ultraviolet radiation. So let me go back. Sometimes when there's two T's in a row in the DNA, just by chance, the two T's, they're bonded to the two A's across from them, but they can bond together like that. And we call that a thymine dimerization. That's a common mutation, a thymine dimerization. It's caused by ultraviolet radiation. Another common mutation is, look, there's a T. What's supposed to be attached to it? An A. But look, a G is attached there instead. And what that does is, remember, G is supposed to go to C. Well, that makes a kind of a, it doesn't attach right, and it kind of sticks out on both sides, and that's why they got this kind of bump in there. Now normally, now this is actually happening when your cells copy one another, you get mutations all the time. But you have enzymes that are repair enzymes. And they go through the DNA, and they're basically moving along it, feeling it. And if they move along right here, they feel a bump. It's sticking out because it didn't bond correctly, because that's not a C, it's a T. And so this sticks out there, and this sticks out there. And so a repair enzyme crawls along the DNA and feels a bump and goes, hey, we got a problem here, and it'll actually remove the base and add the right one in. And this causes the mutations to be a lot lower. Normally about 1 in 1,000 DNA bases is mismatched. But after the repair enzymes come through, it's cut down to something like 1 in 1 million or 1 in 10 million because you're repair enzymes. And that keeps your DNA instructions relatively the same, copy after copy after copy. There's some mutations, but usually not enough to cause major problems. This person has a mutation of the repair enzyme.
That's really bad.
So they don't repair the DNA that's mutated. And that's what they look like. These are all tumors and mutated cells. People with xeroderma pigmentosa, they can't go out in the sun at all. Like a minute out in the sun will cause all kinds of problems. They almost all die at a very young age from cancer that spreads around their body.
Can't they still get mutations without going in the sun?
Can they what?
Can they still get
You can still get mutations without going into the sun. But the sun really causes those thymine dimers and stuff like that. So they especially have to avoid that if they want to live longer. But it's a terrible disorder. You would know if you had it. It's something you're born with. It's a mutation of the repair enzymes. By the way, this is what skin cancer looks like. If you ever have weird moles that are dark or discolored or not shaped in a nice circle like a normal mole is a circle, if they're shaped weirdly or something, you need to get that checked out by a doctor. Often it occurs on places you don't look at very often, like your back. And so sometimes they can get kind of out of control before you realize it. Cancers like this can spread very quickly. So check yourself for moles and things. There are problems with the DNA. Your skin doctor, if you go to a skin doctor every once in a while, they make you take off your clothes, they look at your whole body, checking you out. It's kind of weird, but you'll probably live longer if you get that done. Okay, so I just kind of ran through the whole end of the chapters. If we have a quiz tomorrow, it could be over either 14.3 and 4 or 14.5 and 6, or both. Boo! Woo!